All right. Well, I love exercise physiology and muscle physiology. So I'm super excited to keep going on chapter 12. First, so so this is part 2, by the way. Let's review a little bit in case you watched part 1 a while ago. It's worth reviewing cross-bridge cycles. So again, this is happening in the sarcomeres. So each sarcomere is undergoing the cross-bridge cycle as long as the motor neuron is stimulated. And we'll get to that excitation contraction coupling. So excitation contraction coupling is really combining the cross-bridge cycle with excitation of the lower motor neuron. So we have the actin strand, which is also known as the thin myofilament. And then we have the myosin strand, which is also known as the thick myofilament. And so it's really an interaction between the myosin heads and the actin strands that are producing a shortening effect in the sarcomeres. And this is cyclical, meaning a myosin head can reach across, attach to an actin strand, pull the actin strand, then detach, and then reattach again. And this is going to occur as long as there is ATP available and the motor neuron is being stimulated and calcium is out in the sarcoplasm and available to bind to troponin. So hopefully this is sounding familiar, but let's go through the steps. So what happens first is that ATP is going to bind to a myosin head, and the energy from the ATP molecule is going to be transferred into the myosin head. And so it's said to have the myosin head, that's head, is energized. We just added potential energy from the ATP into the myosin head. It's now energized. And what that does is it puts the myosin head in a conformation. Once it's energized, myosin head is in the right conformation or shape to bind to actin. So as you see, this is energized, and that myosin head is now has energy in it, and so I'm starting here because binding the ATP is going to energize the myosin head. It goes into the high energy form. When it's in the high energy form, it increases the affinity for myosin head to bind to the active site on the actin strand. So it's going to reach across and it's going to bind here. And this right here, when the myosin head is attached to the actin strand, is called a cross bridge. A cross bridge is formed. I always abbreviate it to look like that. So a cross bridge is formed when the myosin head is attached to the actin strand. That is the definition of a cross bridge. When that occurs, the minute a cross bridge occurs, the binding of the myosin to the actin strand, which is a cross bridge, the inorganic phosphate is released off the myosin head. This is going to activate a power stroke. So inorganic phosphate falls off, activates the power stroke. What is a power stroke? A power stroke is taking the potential energy that was in this myosin head and turning it into kinetic energy. That means it's actually moving the actin strand. So a power stroke is where the myosin head releases stored energy and moves actin strand. That's a power stroke. Actin gets pulled towards the middle of the sarcomere due to the power stroke. And at that point, ADP is released off the myosin head. Now, a new ADP, a new ATP has to bind to the myosin head in order to decrease affinity of myosin head to actin strand. And that causes the release of the myosin head from actin strand. And then we start the whole cross bridge cycle over, and we're back to ATP is going to then be consumed, so to speak. The energy from the inorganic phosphate bond is going to go into the myosin head, and we're going to start this all over again. That's what ATP is hydrolyzed means. It means the energy from the ATP bond is being put into the myosin head. Myosin heads have an enzyme on them that causes this hydrolyzation. And if the enzyme is really fast to take the energy out of the ATP and put it in the head, it's called a fast muscle fiber. So fast twitch and slow twitch muscle fibers are dependent on that enzyme activity in the myosin head. Another thing I want to mention is these little green molecules right here. These green molecules are calcium. And if calcium is bound to the molecule troponin, then this strand here called tropomyosin rolls off the active sites of the myosin heads. So calcium's bound to troponin. It's going to move tropomyosin off active sites, so cross bridge can occur. So again, we have to have calcium present in order to have the active sites open so myosin heads can reach across and form a cross bridge with the actin. And we also have ATP. We have to have ATP available. What causes calcium to be available? Oh, sorry. I'm going to move on, but this is a video for you to watch to review. So excitation, contraction, coupling, this is where we're coupling how the cross bridge cycles are turned on and off. So when it says how muscle contractions are turned off, it's really how does the cross bridge cycle get turned on and off? And it's kind of what I was talking about, is that we have a neuromuscular junction. So each muscle fiber has one neuromuscular junction. And so each muscle fiber is going to receive input from a single motor neuron. And this is at the neuromuscular junction. So there's going to be nicotinic cholinergic acetylcholine receptors, and they're going to be located on the motor end plate. And this is where acetylcholine binds to cause an action potential to be generated in that muscle fiber. Motor neurons always create an action potential because it's a nicotinic cholinergic receptor. They're always excitatory. Roles of calcium. As stated before, calcium is a signaling molecule. If no calcium is present in the sarcoplasm, which is really the cytoplasm, then no cross bridges can form because troponin holds tropomyosin over the myosin binding sites. If calcium is present, i.e., if calcium is released from this sarcoplasmic reticulum, then calcium can bind to troponin, causing the movement of troponin off, moving tropomyosin and exposing the binding sites or the active sites for myosin to bind to actin, forming cross bridges. And now you can have cross bridge cycle. It can occur. Steps of excitation, contraction, coupling. Well, an action potential needs to be generated in the sarcolemma. Then that action potential will travel down the transverse or T-tubules. The DHP receptors of the T-tubules will open. These are really voltage gated. So voltage gated calcium channels in the sarcoplasmic reticulum. So calcium can be released into the cytosol or sarcoplasm. Then calcium can bind to troponin, moving tropomyosin off the active site for myosin head to bind. Then cross bridge cycle can occur. It kind of looks like this. So you have the excitation, contraction, coupling. You have an action potential arrive at synaptic terminal of a lower motor neuron, because this is the neuromuscular junction. So action potential arrives, and remember, voltage gated calcium channels will open and calcium comes in, right? Change of voltage, action potential. Opens voltage gated calcium channels, calcium comes in, and then causes exocytosis of acetylcholine, which is the neurotransmitter. These are these little purple circles. Acetylcholine is now in the synaptic cleft, and you have these acetylcholine receptors. These are ionotropic. They are nicotinic cholinergic receptors on the motor end plate of the sarcolemma, which is the cell membrane of muscle fiber. So motor end plate has the nicotinic cholinergic receptors It's highly folded and has a lot of receptors, and acetylcholine is going to bind, and it's always excitatory. Therefore, it's going to generate an action potential in the skeletal muscle fiber. So action potential is generated in skeletal muscle fiber when acetylcholine binds to the nicotinic receptor on the motor end plate. Now we have an action potential that has been created, an electrical current, and it's going to travel down the T tubules. T stands for transverse tubule. So this, now we have an action potential or an electrical current traveling down the transverse or T tubules. At the lateral sacs, where the T tubule meets the sarcoplasmic reticulum. So this is the sarcoplasmic reticulum. It's going to open these channels, voltage-gated channels, and calcium's going to be released. So the action potential triggers calcium being released from the sarcoplasmic reticulum. As long as action potentials are coming down the T tubules, calcium's being released. Calcium is now released, and you can start the cross bridge cycle. So here's the cross bridge cycle. Because calcium now binds to troponin, moving tropomyosin, and now myosin heads can bind. We have multiple myosin heads on the thick filament or myosin strand, and the amount of calcium that's present and bound to troponin is going to determine how many active sites are available for myosin heads to bind. And so it really comes down to the number of action potentials arriving at the neuromuscular junction is going to determine how many action potentials are generated in muscle fiber. Now, how many action potentials are generated in the muscle fiber is going to result in how long voltage-gated calcium channels are open, which determines how much calcium is available to bind to troponin on actin strand, right? So if all of the troponin have calcium bound to it, all active sites are available. For many myosin heads to bind. That is to say, the amount of calcium released is going to equal how many cross bridges can form. And how many cross bridges can form determines how much force a muscle fiber can generate. That is to say, number of cross bridges formed in a muscle fiber equate to how much contraction that muscle fiber can create. Again, here's another video if you want to review. And so we're going to get into contractions. There's something called a twitch. We'll talk about the twitch. And then we're going to talk about factors that affect force generation in individual muscle fibers. Well, I just reviewed that for you, but we're going to go over it again. And then we're going to talk about how we create force in a whole muscle. And then we're going to talk about the speed of which that muscle can produce force. So let's start with a twitch. It's really only seen in a lab. It's one muscle fiber's response to one single action potential. This doesn't occur in kind of real life, but we can stimulate this in a lab. And it can be defined for a muscle fiber or a whole motor unit, which is one motor neuron, and that's a lower motor neuron, and and all of wow, I'm really struggling already. It's going to be a long lecture. All of the muscle fibers that have a neuromuscular junction with that motor neuron. So this is what a motor unit is. We will get into that in detail as well. So let's talk about twitches. There's something called isometric. Iso means same, and metric means length. So an isometric twitch or contraction, the muscle fiber or the sarcomere stays the same length. Sarcomeres stay at the same length. So muscle doesn't shorten. This is generally due to the fact that the load is too heavy, and so the muscle can only contract so much and shorten so much. You only have so many myosin heads that can attach, so many cross bridges can form, or you just want to keep your bones in the same position. So this would be like postural muscles. So isometric, load dependent. Nothing's going to move um, because there's not enough cross bridges formed. You have to have more cross bridges formed than the actual load to move that load. And if you can do that, then you can have what's called an isotonic twitch, which is iso is same, and this is really tone or force or load. And so that would an example of that is like, dumbbells. You have a 10-pound dumbbell, that 10 pounds does not change, right? You can be shortening and lengthening your muscle. It's going to be the same force or load. If you can form enough cross bridges to overcome that 10-pound weight, you're going to move it. And so it's weight dependent, right? If you have, a 100-pound dumbbell, probably not going to be able to actually move that dumbbell. So it's going to become an isometric twitch because you're just contracting, but nothing's moving. So understand that the shortening of a fiber cannot begin until enough cross bridges have occurred or formed. What determines that? Well, you just keep stimulating that upper motor neuron, or and that which stimulates the motor neuron, which then is going to cause an action potential in the muscle fiber, which is going to cause release of calcium. And if you do it over and over and over and over, more calcium builds up, more calcium binds to troponin, more active sites are exposed for myosin heads, more myosin heads reach across, form cross bridges. And when you have enough cross bridges, you can start to move that load. And that's why heavier loads have a longer period of time, because there's a longer period of isometric contraction where you're building up tension, you're building up the number of cross bridges in order to actually move the load. We have to keep forming cross bridges until we can actually move that load. So it's showing you a single action potential. If it's a light load, it's going to take a little bit of time to form cross bridges, right? So enough cross bridges formed. So it takes a little bit of time, and you can start to move that load. And then there is a decrease in the shortening because you move the load, right? If it's an intermediate load, now we're taking a little bit more time here to form cross bridges. If it's a heavy load, we're taking even more time to form cross bridges before the load is moved. But we can only shorten so much when the load is heavier and heavier. And so this is kind of what this is showing you is then uh, if you have a relaxed muscle, you have electrodes set up, and then you contract the muscle, you're going to see tension and then time release because the twitch ends and then the muscle returns to its resting length. Isotonic twitches, there's a constant tension. And when the tension is greater than the load, by the way, tension is created by muscle fibers, i.e., cross bridges being formed. The load is what the muscle needs to overcome, right? So, um, need enough cross bridges to be stronger than the load. So load's going to lift when there's more cross bridges and more force being generated or more tension being generated by the muscle fiber than the actual load. And so this is kind of showing you here we got the muscle shortening and you get tension and then that twitch ends and the muscle's going to lengthen again. And this is load dependent, right? So if it's a 5 kilogram or 5 gram load, that twitch is going to produce enough tension to move 5 grams. If it's 10 grams, it has to produce enough tension for 10 grams. And you'll notice that this period is going to get a little longer when the load increases. So like 15, we have to produce 15. And the latent period gets longer as the weight gets heavier. At 20 grams, though, we can't move it. So it becomes an isometric contraction because there's not going to be any shortening. The load is too heavy. So twitch contractions, not all muscle fibers have the same twitch time, meaning how fast can generate cross bridges and myosin heads can move. So different muscle fibers have different ability to generate cross bridges and to make the myosin heads move. And so the two different factors are one, how much time does it take for the calcium to be released into the cytosol enough to create cross bridges? So how fast is that calcium ATPase? It's going to be very fast in fast twitch muscle fibers. It's not going to be fast in slow twitch muscle fibers. This is to say calcium gets released fast and taken back up into the sarcoplasmic reticulum fast in fast twitch muscle fibers. And it's slow release from sarcoplasmic reticulum and flow to be taken up in a slow twitch muscle fiber. So How fast it can get calcium out and how fast it can get calcium back in is a speed factor, and then also the length of time a muscle fiber takes to form a cross bridge and to do a power stroke. So how fast can do a power stroke, and that has to do with the enzyme on the myosin head. I should say on myosin head. So in a fast twitch muscle fiber, it really has fast enzymes. And so those fast enzymes can energize myosin head quickly. And so it's going to power stroke quick. And so opposite for slow twitch. So the myosin head, how fast it can move and do a power stroke is dependent on that enzyme. And so if it's fast twitch, it's going to have fast ATPase and it's going to power stroke really fast. So factors that affect the force generated by an individual muscle, right? I shouldn't have written that. Now we're talking a whole muscle, whole entire muscle is going to be really determined by three factors. One is how much tension or force can be produced in a single muscle fiber. So, how much force can be produced in one single muscle fiber? What determines that? It's determined by the number of active cross bridges formed. How many cross bridges are formed in a single muscle fiber? What determines this? The frequency of stimulation. So is determined by frequency of stimulation of the lower motor neuron. So if it's a high frequency of stimulation of the lower motor neuron, it's going to have a high number of action potentials created in the muscle fiber, which is going to result in more calcium being released from the sarcoplasmic reticulum, which is going to have more active sites on the actin strand exposed, which means more myosin heads can bind, which means more cross bridges formed. More cross bridges formed, more force in the muscle fiber. Then we can go to the number of fibers that are available in a motor unit. Is it a large motor unit? It's going to have lots and lots of muscle fibers. And if each of those muscle fibers have a whole bunch of cross bridges, then you're going to have a lot of force being produced. And then your central nervous system can decide how many motor units are activated. So let's go through that. So factors affecting force generated by individual muscle fiber, right? So that's just one cell. Frequency of stimulation is going to increase the tension. Frequency of stimulation of the motor neuron. That's going to result. So if you have an increase in stimulation, it's going to increase the tension because more and more calcium is being released, right? So I say, why? Well, more action potentials arrive at the sarcoplasmic reticulum. It's going to equal more calcium released, which equals more sites for myosin heads to bind, which is going to equal more cross bridges, right? So the more calcium's out there, the more cross bridges form, and that is due to the frequency of action potentials arriving at the sarcoplasmic reticulum. So the twitch, um, you can read through this. Um, Loads can change. We have something called torque, and we could get into biomechanics, but we're not. And that we can have the same load, but we can vary the speed of which our muscle's moving because the load can move closer to the fulcrum, and that can increase the speed. And these are all biomechanical things that we're not going to get into. But when it talks about load changes as limb position changes, that's something called torque. That's just a force. Uh, Graded muscle contractions depend on two factors, and what graded muscle means is varying the amount of force, right? So we start off with a little bit of force and we get a medium, and we can keep adding the amount of tension or force produced by each muscle fiber. And so the number of active cross bridges is is going to be a factor in determining how much force. And we could start off with a little bit of force in a muscle fiber, and we can actually make it more and more and more by just adding more cross bridges that are being formed. Because the more cross bridges that are formed are going to equal more force in that muscle fiber. And then we could talk about how many muscle fibers are contracting, and that's really determined by the size of the motor unit. We can also talk about number of muscle fibers contracting, and that lifting heavy weights like weightlifting actually increases the number of myofibrils in a muscle fiber. And you're saying, okay, big whoop. Well, if you increase the number of myofibrils, guess what? You increase the number of possible cross bridges available. And if you increase the number of cross bridges available, now you've increased the amount of force possible in that muscle fiber. So that's why lifting heavy weights, you have your muscles look bigger because you have more myofibrils. More myofibrils mean more actin and myosin, which means more cross bridges, which means you could produce more force. Frequency tension relationship, because a single action potential in a skeletal muscle fiber only lasts for a couple of milliseconds. That's the action potential. But the contraction or twitch can last for hundreds of milliseconds. And so you can have a second action potential being initiated during the period of mechanical activity. That's just saying you can add more cross bridges while you still have cross bridges formed. And so when a stimulus is applied before the fiber has completely relaxed, then you can add more cross bridges to the existing cross bridges. Right? So the force can get stronger. And so this frequency tension relationship, that is to say the frequency of action potentials generated in the muscle fiber. Well, that's due to the number of action potentials coming from the motor neuron, right? Is going to then determine the amount of tension, which is essentially a number of the amount of cross bridges formed in the muscle fiber. So you can have action potential, action potential, another action potential, another action potential, another action potential, and it's just adding cross bridges, which means the force goes up, right? You have more cross bridges, you have more force. Well, if you are maximally stimulating that motor neuron, then all troponin has a calcium bound, and so all cross bridges available are formed. That's your max. That's your max amount of force, and so that's called a fused tetanus. It just means you've maximally stimulated that motor neuron to the point where you have so much calcium, that concentration is really high in the sarcoplasm or cytosol, that you have maximal amounts of cross bridges formed, and that's going to equal max force. So we call that peak force. So factors affecting individual muscle fibers. The amount of tension developed is based on the amount of calcium that's bound to troponin. And what determines that? Frequency of APs generated in muscle fiber, which is determined by frequency of action potentials generated by the motor neuron. That's going to cause calcium to be released faster than it's being actively transported back up into the sarcoplasmic reticulum. So calcium's released, but also active transported back into the sarcoplasmic reticulum. So you have to have more calcium being released than being taken back up, reuptake back in the sarcoplasmic reticulum. And if you're stimulating that motor neuron, you're creating an action potential that is going down the T tubules and releasing calcium out of the sarcoplasmic reticulum faster than it's being pumped back in, you're eventually going to have all troponin have a calcium bound to it, which means you can have the max number of cross bridges, which means you have the maximum force of a contraction, which is peak force in that muscle fiber. And so we see that there. Fiber length matters. If we want to get max cross bridges, we have to have an optimal length of overlap. So this is the sarcomere, actin, myosin overlap to have max number of possible cross bridges formed. So the resting length at which a muscle fiber can develop the max tension, it has to have the max overlap of actin and myosin. And so if we look at here, actin's already fully overlapped. And so you're not going to have max tension because you can't really go much farther. There's too much overlap. From here to here, this is where max number of possible cross bridges can form. And so that's your optimal overlap. Here, you can't have max. Some of these myosin heads are not overlapping an actin strand. Here, many myosin heads are not overlapping, so you're not gonna have no max cross bridges possible in this scenario. And so fiber length matters. Um, you need optimal overlap. Fiber diameter. This is what I was talking about in number of myofibrils in a muscle fiber. That's really the diameter. How many myofibrils are in a muscle fiber determine the diameter. So if it's bigger in diameter, you're going to have more myofibrils. You're going to generate force because you have a higher number of cross bridges possible, right? So more cross bridges possible, you're going to generate more force. More sarcomeres aligned in parallel, you're also going to generate more force. And so fiber diameters vary in different muscles. Some muscles have a lot of myofibrils in a given muscle fiber, and some are smaller. And so what we see is if you have a larger diameter, that just means there's more myofibrils there. That's what makes it bigger, which means you're going to generate more force. And that's what, again, weightlifting. You are trying to increase number of myofibrils, which increases diameter. They just get bigger. You don't increase the number of muscle fibers. You increase the number of myofibrils inside that muscle fiber, and you are increasing the diameter, which increases cross bridges, which increases force. That's in a muscle fiber. Now we're talking about an entire muscle. And entire muscles have motor units. And so by recruiting motor units, you can vary the amount of force, right? So that's called recruitment. More muscle fibers contracting is going to increase the overall muscle tension. How do you do that? You stimulate more motor neurons, which then stimulate more muscle fibers. And motor neurons are set up as motor units. You can have motor neuron X, and motor neuron X innervates these red guys. So 1, 2, 3, 4, 5. So it branches to 5 red muscle fibers, just in this. And then you have motor unit Y. And motor unit Y goes to 1, 2, 3, 4, 5, 6, 7. So is motor unit X bigger or smaller than motor unit Y? Well, X is going to be smaller because it only has 5 fibers, muscle fibers. Motor unit Y is going to be bigger. It has 7. So if you want to have some force, you just activate motor unit X and you have those fibers start to generate cross bridges and contract. If you want more force, you can activate motor unit X and motor unit Y, and now you have more force. Why? Because you have more cross bridges, because now you are using 12 muscle fibers cross bridging at the same time. And so that's what recruitment is. You're recruiting motor units. You're recruiting the motor neuron and all of the muscle fibers it innervates. So the definition of a motor unit is the motor neuron and all of the skeletal muscle fibers it innervates. And so if that motor neuron is excited, it's going to generate action potentials in all of the muscle fibers. It forms neuromuscular junctions with. And some muscles have hundreds of motor units, and some muscles have a few motor units. And that's going to depend on the size and the function. Like your eye muscles, quick movements, very precise. We have, only a few motor units, and they're small motor units. But in your quadriceps, you're going to have a lot of motor units, and they're going to be big motor units. So within a muscle, it's going to vary in the number of motor units that are there. But we regulate force in a whole muscle by motor unit recruitment. And so the more fibers recruited, the greater the tension. And so we just recruit motor units one at a time. So the activation of a motor unit is going to activate all the muscle fibers in that motor unit. And then we can increase the tension in that motor unit by increasing the frequency of action potentials to that motor unit, right? Again, small motor units are for fine, very controlled movements. So we're going to have small motor units exclusively in areas where we really want to control the amount of force. Large motor units are going to be for gross, large muscle movements. So your quadriceps or glutes, they're just doing big things. They have one motor neuron going to hundreds of muscle fibers. So motor unit size is determined by the number of fibers that are attached or innervated by that motor neuron. And it's going to vary, right? Small motor units are for delicate movements. Large motor units are for big, strong movements. Fiber diameter is going to vary in motor units. So you can have small diameter motor units. Those are for very gentle movements. And you can have large diameter. And we're going to see that in a second. One thing I have to say is that with motor units, when we get to, say, fast twitch or slow twitch, the myosin ATPase is same in a motor unit. So if it's a fast twitch motor unit, they all have fast myosin ATPase. If it's slow twitch, they all have slow. So here's the size principle. Almost all skeletal muscles have all three types of motor units. They're going to have the small, they're going to have the intermediate, and then there's going to be the large. The amount of tension developed by each fiber is really determined by size, right? Number of possible cross bridges. And that's really determined by the number of myofibrils. And then the number of fibers and the type of fiber is the next thing. Are they fast twitch? Are they slow twitch? And so forth. We recruit based on size. So we're always going to recruit the small motor units first. They're smaller in diameter. They're easier to excite that motor neuron. It's easier to get those fired. When I say fired, I mean firing an action potential, making the muscle fiber contract. The intermediates take a little bit more work to generate action potentials in those motor neurons, and then the large ones take a lot more work. And so we recruit based on size. Smaller motor units first. And remember, activation of a motor neuron activates all the muscle fibers that are in that motor unit. And so an increase in tension occurs in a stepwise fashion from small to bigger. So small motor units first, intermediate, and then large. Order of motor unit recruitment, again, X is the smallest. It innervates only a couple muscle fibers, but it's really easy to generate an action potential. As they get larger, it's more difficult for that motor neuron to reach threshold. And so we see that small motor units just are easier to generate, right? I should say easier to generate action potential in that um, motor neuron. And so you're going to activate those muscle fibers easier. So you're going to recruit those first. If we need more tension, our nervous system works a little bit harder to recruit or activate the intermediate. And then if we really want peak force, we work real hard to recruit the large motor units. And so this is coming from the brain. The upper motor neurons have to work harder in those big motor units. And it's interesting, not sure exactly why, but it seems that have to work harder to create enough graded potentials in the large motor neurons in order to reach threshold. And so there's this recruitment based on size. Velocity of shortening, how fast can you shorten? Really depends on how long the latent period is, right? So the latent period of shortening increases with increasing the load. Larger loads take a lot longer to move than short loads or small loads. So the duration of shortening is going to decrease with decreasing load. That means takes less time to move a small load. We can do it faster. So the velocity, which is the speed of shortening, is going to increase with decreasing load. That is to say, inversely proportional. As load gets heavier, more cross bridges need to form to overcome load, and that takes time. So when we say the velocity is inversely proportional, that means small loads can move quickly. Large loads move slowly. So time and velocity are inversely proportional. As one goes up, the other goes down. So with zero load here, you can start moving it right away. So here's time. At time zero, if there's no load, you can shorten. This means shorten. If it's a little bit more load, there's a little amount of time, right? So say like 5 seconds, milliseconds, to start moving, right? Distance shortening means moving the load. If it's a really heavy load, though, it's going to take more time. Why? More cross bridges need to form, and that takes time. So the latent period increases with increased loads, which equal increased time, right? Which means the speed's slower. So this inverse relationship, this is really what the load velocity relationships look like. Because at some point, if the load gets too big, instead of shortening muscle, it starts to lengthen because the load is too heavy. So when we look at this is the velocity, this is speed, right? So as we go up, this is fastest and this is slowest. If we have no load, right? So this is zero load, it's maximum. We can go fastest, right? So maximum shortening velocity is happening at zero load. As we increase this load, the velocity is going to be medium, right? Because it has a latent period to form enough cross bridges to move load. Load gets even heavier, it's getting even slower. The velocity is decreasing. Here is maximum isometric tension. You can hold this. You have enough cross bridges to hold. But no speed, not moving, isometric. If you keep increasing the load, now your muscle is just like getting longer or it has to drop the weight. And so the velocity is like negative, you're going in the opposite direction. So load velocity relationship is shortening velocity is determined by the rate at which individual cross bridges can keep going, right? How fast can you cross bridge? And that has to do with the rate of the ATP. So fast twitch has fast ATPase, which means it can move faster. It can move the load a little bit faster. So faster velocity. So different muscle fiber types also have a different velocity. As load increases, it's slowing the cross bridges and slowing the power stroke. And so it slows down that rate and it's going to slow down the speed of which it can shorten. All right, last thing we're talking about really is muscle metabolism and how muscle cells generate ATP. I keep talking about fast twitch and slow twitch. That is the ATPase speed on the myosin head. But there's also how muscle cells generate ATP because we need ATP, right? You can have that motor neuron being stimulated, but if we don't have ATP, that muscle fiber is not going to cross bridge. So what are our sources of ATP in muscle fibers? We have creatine phosphate on tap, just laying around in the muscle fiber. Then we could do anaerobic glycolysis. This is glucose to pyruvate, and we get 2 ATP per glucose. Then we have oxidative phosphorylation. That's when we use the mitochondria, and here we can use triglycerides and glucose and generate lots of ATP. So these are our 3 ways. And so if we look at this, right, we have stored glycogen in muscle fibers, and that can be turned into glucose. And oops, what did I just do? And we can get 2 ATP per glucose. If we have oxygen, then we can go Krebs cycle, electron transport chain, oxidative phosphorylation, and make a lot of ATP. If we know oxygen, then pyruvate has to reduce NADH, and it becomes lactate. And so lactate is going to decrease pH, increase acidity, and we are going to slow down. All right. So we have 3 ways muscle fiber can generate ATP. We have substrate level phosphorylation of creatine phosphate. We have substrate level phosphorylation via glycolysis. And we have oxidative phosphorylation in the mitochondria. And here's where we can use fats and sugars. And here's where we use creatine phosphate. And so creatine phosphate is a one-step enzyme reaction. So you get ATP instantly, but you just don't have enough creatine phosphate to go forever. But it does get you to the point where glycolysis is now producing ATP. So we're going to first use creatine phosphate. We use that while we get glycolysis up and running to create ATP. And if there's oxygen available and we are able to start using fatty acids through beta oxidation, those fatty acids are mass producing ATP. And mass production. So it's going to kind of look like this. So the role of creatine phosphate, it's quick, right? It's a quick on tap ATP synthesis. Doesn't last forever, totally reversible. And so it can only supply small quantities of ATP. And so what happens is we use creatine phosphate first, right? So this is our energy source. In the first 1 minute, we're using creatine phosphate predominantly. But in that first minute, we are also starting to do glycolysis. And here is where we are doing more glycolysis and less creatine phosphate. But that's okay, because if we keep going, we're also, as there's oxygen present, starting to also use oxidative phosphorylation of triglycerides and glucose. And this is occurring in about 2 to 5 minutes. If we have oxygen, we're going to start doing more oxidative phosphorylation and less glycolysis. So more and less glycolysis. This is good because remember, glycolysis only gives you 2 ATP. Oxidative phosphorylation gives you anywhere from 34 to hundreds of ATP, depending on what source, if you're using fats, triglycerides, or glucose. So this is kind of why the first 5 minutes of any cardiovascular exercise is tough, because you're using glycolysis, you're not making really much ATP until you cross over into using more oxidative phosphorylation. And then you're really producing ATP super efficiently, and you feel better because you have more ATP on top, you have more energy. This is for low intensity. High intensity, we're using creatine phosphate. We have the same 1 minute. We don't have oxygen available, so we're doing anaerobic glycolysis, and we are burning through ATP and converting pyruvate to lactate or lactic acid. So we have a buildup of lactic acid. Even though oxidative phosphorylation, it never makes it there because not enough O2 available because you're going too fast. And so we're not getting enough oxygen. We're creating lactate, and we can only do high intensity for so many minutes. So muscle cell metabolism with exercise intensity, if it's light to moderate, right? We're initially using creatine phosphate and then glycolysis. But after about 5 minutes, it's mostly oxidative phosphorylation, and we're using really triglycerides, lipids, fats. So we're getting a lot of ATP. And we can keep going and doing this for long periods of time as long as we have good ventilation. And as long as our heart rate and contractility is good, we dilate the vessels to the muscles, and we also are increasing the number of GLUT4 channels in the muscle fiber to increase amount of glucose that gets into the muscle cell. So all of these are happening with light exercise. This is why light to moderate intensity exercise is really good for diabetics because light to moderate exercise increases the number of GLUT4 channels, and it increases facilitated diffusion of glucose into muscle fiber. This helps control blood glucose levels without insulin. So no insulin is needed. So exercise is really good for diabetics. When the exercise intensity goes up, lactate or lactic acid builds up. Why? Because pyruvate needs to oxidize NADH back to NAD, so at least you can continue with glycolysis, but pyruvate then becomes lactate with those extra electrons and protons, and this is an acid. Also not generating much ATP with glycolysis. And so muscle fatigue is after 5 minutes. Creatine phosphate, which has been used, can you use that phosphate, but it can actually convert back to creatine phosphate. When you stop, you breathe and increase O2 back, and then lactate converts back to pyruvate, and pyruvate can now go through Krebs cycle and oxidative phosphorylation, et cetera, et cetera. So that's why we have to stop. We have to breathe. We have to kind of reset the system. Muscle fatigue is when a muscle fiber is repeatedly stimulated by motor neuron, but the tension in the fiber eventually is decreasing, even though you're still stimulating it. So why? Muscle fatigue, which is a decline in muscle tension, it's really a decline in the number of cross bridges formed. Why? Because you ran out of ATP or well, I should say the velocity decreases. It's also a slower rate of relaxing. Onset of fatigue is really developed by the type of muscle fiber. If it is a fast glycolytic, it only can do glycolysis. So it's going to burn out real quick. Fast oxidative glycolytic will take a little bit longer. And so that's where we get to the intensity and the duration is how much O2 is available and also what type of muscle fiber, because fast glycolytic can only do glycolysis. They will burn out. And then also the degree of fitness, muscle fibers can be trained. Meaning you can increase glycolytic enzyme. So you can do glycolysis longer. You can increase the ability to tolerate lactic acid. So there are training adaptations. So muscle fatigue causes two things: conduction failure. That is where the muscle fails to produce an action potential in the muscle fiber. So no action potential in the muscle fiber, no release of calcium out of the sarcoplasmic reticulum. This conduction failure actually results from a buildup of potassium ions during the repolarization phase due to repetitive action potentials. So that's a conduction failure. You cannot conduct another action potential. An increase in external concentration of potassium is going to lead into a more difficult time generating an action potential in the muscle fiber. That's one. The second is lactic acid buildup. As the amount of lactate gets built up, it increases the concentration of protons, which is another way of saying it decreases the pH and increases the acidity. And that is going to kind of make it harder for the enzymes. Enzymes don't do well in acidic environment. So when the acid builds up, enzymes don't function properly. And if the enzymes don't function properly, you're not going to be able to produce ATP. And so anything that's powered by ATP is going to have a harder time functioning, like the ATPase, the calcium ATPase pump. And so it's going to impair not only the ability to contract, but also the ability to relax. So it's basically muscles spasm or cramp. We call it muscle cramping, where the muscle just contracts and cramps because you can't pump that calcium back into the sarcoplasmic reticulum. That's another type of fatigue, is that cramping. And that's really due to the lactic acid buildup and the enzymes not functioning properly. Types of muscle fiber types, I keep talking about fast twitch, slow twitch, so I'm going to define those now. Fast twitch, slow twitch are based on the velocity or speed of the myosin heads to pivot. How fast is the power stroke? And then the second thing is, how does it generate ATP? So how fast are the myosin heads and how they generate ATP? And this is how we classify muscle fibers. So fast and slow fibers contain different myosin ATPase activities. Fast twitch has fast myosin ATPase, which equals fast or high velocity power stroke. Slow as slow. And so it's going to be slower to contract, and I should say both of them relax as well. And so that determines the rate of the cross bridge cycle. And so that determines the speed of the muscle fiber shortening is that ATPase activity. So rate of myosin ATPase activity, ATP, how fast can you create ATP and use that ATP? So fast has fast ATPase activity, slow has slow. Then there is how you create that ATP. And each muscle fiber has a mixture of fast and slow muscle fibers, but the proportions vary. So, like, how many of them are fast versus slow can vary, right? So some muscles are dominated by slow twitch fibers and some muscles are dominated by fast twitch fibers. In a single motor unit, all of the muscle fibers are the same type. So if it's a fast motor or fast muscle fiber, the motor unit's all fast or all slow. So we have slow oxidative fibers in motor units, wide, slow, myosin ATPase, and calcium ATPase activity. Also produces ATP via oxidative phosphorylation. So mass produces ATP. These get recruited first. Why? Easier to excite and produce ATP continuously. And that matters, right? Because you're going to be using them over and over and over and over and over again. So you need to be able to produce enough ATP. Then you have fast oxidative fibers. These have fast myosin ATPase. So they move fast and produce ATP via oxidative phosphorylation or glycolysis. So those are the fast oxidative. A lot of times they're fast oxidative glycolytic or FOG. And then you have the fast glycolytic. These are fast, but only can produce ATP via glycolysis. They are anaerobic to ATP per glucose. So don't produce a lot of ATP, generate a lot of lactic acid. Now, the types of muscle fibers, slow oxidative, they look red. So sometimes they're referred to as red fibers. That red is due to a pigment called myoglobin. It's a protein in the muscle fiber that binds oxygen. Like hemoglobin in a red blood cell, myoglobin binds oxygen in the muscle fiber, so keeps O2 around on tap. That is to say, slow oxidative fibers store a little bit of oxygen. So they're always going to have oxygen available. They have a lot of capillaries, so they have a lot of blood flow to them, and they have myoglobin that binds that oxygen, has oxygen supply on tap. Fast oxidative glycolytic, they're called pink fibers. They're not quite red. Why? Because they have some myoglobin and okay capillary blood delivery. So they can still do oxidative phosphorylation. Then we get to the fast glycolytic. These guys, we call these white fibers. No myoglobin. So they don't really have very much oxygen, and they are doing glycolysis or anaerobic glycolysis. So comparison of fast and slow fiber contractions. Fast fibers contract 2 to 3 times quicker. They have a 2 to 3 time higher velocity of the power stroke, pulling that actin over the myosin. And they have a higher calcium ATPase. So they can start faster and stop movement faster. So these enable athletes to start a contraction quickly, but also stop a contraction quickly, which can matter. If you're playing basketball and you need to stop moving in one direction and turn to move in the other direction, you also want to be able to stop your muscles quickly as well. So fast fibers are good for starting and stopping. And so they're going to be quick. The intermediates, like your gastrocnemius, has a high number of fast oxidative glycolytic. They're medium, and then your soleus is mainly slow oxidative. So these are fast oxidative glycolytic, and these are fast glycolytic. So how fast do we want our fibers to move, and how fast we want those muscles to go and how we train them are really going to determine, you know, how fast we are at doing certain things. We can train our muscle fibers to be more oxidative by just doing more oxidative type training. Oxidative fibers, these are your slow oxidative, slow twitch. They do oxidative phosphorylation, mass ATP production. They have a lot of mitochondria. That's what helps them mass produce ATP. They have myoglobin, stored O2 available for oxidative phosphorylation. They have many capillaries, so high oxygen delivery system. They're small in diameter, so they're easy to activate or recruit. And they're resistant to fatigue because continuously producing ATP via oxidative phosphorylation. So you don't have lactic acid buildup. Glycolytic fibers, these can be your fast oxidative glycolytic or fast glycolytic. These are your type 2s. They do anaerobic glycolysis. They can be trained to do oxidative phosphorylation. These really are your intermediate fibers. We can train these to be more oxidative or more glycolytic. Not a lot of mitochondria. They're not producing ATP via oxidative phosphorylation, but they do have a lot of glycolytic enzymes. Training increases these. Remember the podcast, can training change your DNA? right? So they showed that training can increase glycolytic enzymes. You can store more glycogen, so you have more glucose on tap. Not a lot of capillaries because not using O2. They're large in diameter, so we recruit these last because they burn out quick. So we're not going to use those. They're quick to fatigue. We're not going to use these unless we need a whole lot of force to be generated. So this goes through slow oxidative fibers, also called type 1 fibers, slow twitch fibers, or red fibers. Uh, Low myosin ATP, they're slower to cross bridge, but they have a high capacity to produce ATP. Fast oxidative glycolytic, they're also called type 2 or intermediate or pink fibers. And they have high myosin ATPase activity, but they can generate ATP. They can be trained to generate ATP through oxidative capacity. You can increase the number of mitochondria in these fibers, or you can increase the amount of glycolytic enzymes. Depends on how you train. And then you have fast glycolytic. They're also called type 2B, fast switch, or white fibers. And these are glycolysis only. They are the last to be recruited. So muscle adaptation to exercise, increase in the amount of contracting activity, um, increase size of muscle fibers, increase the ability to produce ATP. So aerobic exercise, like running, we're predominantly using these type 1, these slow oxidative fibers. We are going to increase the number of mitochondria and increase blood supply. And both of these are going to increase our ability to go longer. Versus anaerobic, weightlifting, sprinting, we're increasing the number of myofibrils in a muscle fiber. We can also increase the amount of glycolytic enzymes. And so training adaptations are really specific to the type of training you're doing. So you can increase the oxidative capacity by increasing the size and the number of mitochondria. You can convert in the fast oxidative glycolytic fibers. You can convert them to be fast oxidative fibers, increase the amount of capillaries, glycogen reserves. This is glucose on tap versus high intensity, uh, not really increasing the oxidative capacity, but you can take those fast oxidative glycolytic and make them fast oxidative. You're not really increasing the number and size, but you're increasing the number of cross bridges. You can increase glycogen reserves, how much glucose you have stored, and you can also increase the amount of glycogen enzymes for glycolysis. You can also increase the ability to recruit these motor units quicker. So sprinters really want to be able to do this, right? You want to be able to start the race as fast as you can. So you want to be able to start using those fast glycolytic fibers immediately. And so you can actually increase the recruitment of them. Hypertrophy is gaining size. So hyper is large, atrophy means without size. So atrophy is from disuse of muscle. So you're not using it. It's a use it or lose it. Any kind of training adaptations you have to training can go away if you do not use them, and that's called atrophy. So atrophy happens if you're not exercising regularly or bed rest or you have an injury, something like that. It can also happen with age. The max amount of force you can produce is going to decline. 30 to 40% between the ages of 30 to 80. That means you can work out at the same intensity and duration, but it's not going to produce the same effects because we're losing our ability, we're losing our myofibrils. So we have to work harder to just keep same number of myofibrils and all those training adaptations, which is not great. Um, Muscle activity across a joint, there are proprioceptors. By the way, muscles can only pull on a bone, they can't push a bone. And we have what's called muscle spindles. They are monitoring the length of the muscle. There are intrafusal fibers and extrafusal fibers. Intrafusal fibers contract the cells of the muscle spindles or receptors that tell brain the length of the muscle. And they adjust the sensitivity, they're adjusted to the sensitivity of the muscle stretch. Extrafusal fibers are responsible for the muscle contraction. And so, oh, guess I didn't mean to do that. As we stretch a muscle or contract a muscle, these muscle spindles are sending muscle length info into brain so the brain can adjust. Or at least so it knows the length of the muscle. Golgi tendon organs, these are in the tendons and they're inside the tendons sensing tendon stretch. So how much stretch and tension on the tendon. And this is also sending this information into the brain. So again, the brain can adjust. This is to protect against overactivity. When you're stretching and you start to feel your muscle or your limb shake, it's really due to these Golgi tendon organs sensing too much stress, and so it's trying to contract your muscle to not overstretch the tendon. So that's why you get the shaking. All right, that concludes muscles, and next we will go on to cardiovascular system.